There are various genetically engineered mouse models to study pancreatic cancer. The most commonly used model is the KRAS P53 mouse model that harbors an activating mutation in the KRAS oncogene and an inactivating mutation in the P53 tumor suppressor gene. Both mutations are conditionally activated by the PDX-CRE promoter. This KRAS P53 PDX-CRE mouse model, in short KPC model, develops endogenous pancreatic tumors between the age of 2 to 6 months. In preparation of your ultrasound procedure, the following equipment should be prepared and ready to use. For high-resolution ultrasound, a stationary ultrasound device is used. The ultrasound procedure is conducted in general anesthesia using isoflurane evaporation. Once the mouse is under deep sedation, it is transferred to the green working stage. After the ultrasound procedure, the mouse should be placed in a preheated cage to accelerate recovery. Tumor development is highly variable in the KPC mouse model. To avoid unnecessary ultrasound screening procedures, we usually perform abdominal palpation weekly starting from the age of 2 months. Gently palpate the mouse abdomen up and down to detect any